In emergency medicine, the first rule of a blood transfusion is strict but straightforward. Match the patient's ABO and Rhd antigens to prevent an acute immune reaction. If a trauma patient with A-positive blood receives an A-positive donor unit, the biological integration is almost always seamless. The donor cells replenish the patient's oxygen capacity, and the patient stabilizes. But the clinical outcomes change drastically for patients who require lifelong blood transfusions for chronic diseases. A patient receiving their 50th transfusion can be given a donor unit that perfectly matches their A-positive blood type, yet their body violently rejects it. Instead of integrating the new blood, the patient's immune system aggressively targets the donor cells. This reaction breaks down the transfused red blood cells, releasing toxic byproducts into the bloodstream and plummeting the patient's oxygen levels. The transfusion intended to save their life actively worsens their condition. For chronically transfused patients, basic ABO and Rh compatibility provides a dangerous illusion of safety. To understand why this rejection happens, we have to look at how blood is categorized. It comes down to antigens, inherited biochemical markers that sit on the surface of your red blood cells. The ABO and Rh systems are defined by highly prominent structural proteins. If you have type A-positive blood, your cells are coded in large A antigens and Rhd antigens. Humans naturally develop antibodies against the ABO antigens they do not inherit. If you have type A blood, you carry anti-B antibodies in your plasma. If type B blood enters your circulation, these antibodies immediately lock onto any cell carrying a B antigen and destroy it. Rh antibodies operate slightly differently. An Rh-negative person does not automatically carry anti-Rh antibodies, but they will rapidly form them if they are exposed to Rh-positive blood. Because the ABO and Rh antigens are so physically large and highly reactive, matching them prevents catastrophic immune failure in the vast majority of single-event surgical and trauma transfusions. The entire global blood banking infrastructure was scaled around this specific biological threshold. Blood is collected, labeled, and refrigerated based almost entirely on these 8 basic types. This 8-type system was designed for the logistics of trauma. For patients who only need blood once, these 8 categories are sufficient, but they are not the only markers the human body recognizes. The human immune system is far more sensitive than the ABO and Rh labels suggest. It recognizes dozens of other distinct blood group systems. Between the large primary markers, red blood cells carry minor antigens. These include systems known as KEL, Duffy, KID, and MNS. These structures are smaller and less immunogenic. During a single surgery, mismatched minor antigens usually slip past the immune system without triggering a crisis. The clinical threat emerges during chronic care, where a patient is subjected to repeated, sustained exposure to foreign minor antigens over many years. This repeated exposure triggers alloimmunization. The patient's immune system slowly learns the exact structure of the foreign minor antigens and synthesizes specific antibodies against them. Alloimmunization is permanent. Once a patient is sensitized to a minor antigen, any future blood transfusion containing that specific marker will be flagged for immediate destruction. Chronic transfusion elevates these minor, nearly undetectable cellular markers into lethal clinical threats. If minor antigens pose such a severe risk to chronically transfused patients, blood banks would ideally match all of them by default. But logistical protocols collide with population genetics. The frequencies of ABO, Rh, and minor antigens are not randomly distributed. They are dictated by thousands of years of evolutionary genetic pressures and geographic population bottlenecks. This chart maps the ABO and Rh blood type distribution across United States demographic groups. O-positive is the biological baseline, standing as the most common blood type across every single population. But look at the B allele. B-positive blood is found in roughly 9% of white and Hispanic donors, yet it appears in 25% of Asian and Pacific Islander populations. The Rh-negative trait is even more isolated. It is present in about 15% of people with European ancestry, but is nearly non-existent, often under 1%, in Asian and African populations. It is overwhelmingly a European genetic inheritance. Minor antigens like Duffy and KEL follow identical evolutionary pathways, bound to specific geographic ancestries. An antigen profile is a strict genetic fingerprint. Because blood is inherited geographically, finding a precise minor antigen match usually requires finding a donor from the exact same ethnic background. We can see the severity of this genetic reality by looking at the clinical population that requires the most chronic transfusion support: patients with sickle cell disease. Sickle cell disease predominantly affects patients of African, Mediterranean, and Middle Eastern descent. To prevent cells from sickling, blocking blood vessels, and causing catastrophic organ failure, these patients require lifelong, repeated blood transfusions. Finding a safe donor match for a highly sensitized sickle cell patient is a mathematical gauntlet. Imagine a pool of 1,000 random donors. First, we filter for an exact ABO match. The available blood supply drops immediately. We add the requirement for an Rh match. The pool shrinks again. Now we filter for the KEL minor antigen, which the patient has become sensitized to over years of transfusions. Viable options vanish. Finally, we apply the filter for the Duffy antigen. Because the donor pool lacks the specific ethnic alignment of the patient, the available blood supply drops to zero. For a patient population requiring chronic transfusions, a donor pool that lacks genetic diversity carries a lethal mathematical certainty. Chronic transfusions aren't the only event where minute antigen differences cause irreversible immune sensitization. In prenatal care, a similar threat occurs when an Rh-negative pregnant person carries an Rh-positive fetus. If fetal red blood cells cross into the maternal circulation during pregnancy or delivery, the pregnant person's immune system detects the foreign Rhd antigens and begins producing anti-D antibodies. Once sensitized, the pregnant person faces a severe risk in any subsequent pregnancy with an Rh-positive fetus. The maternal antibodies will actively cross the placenta and destroy the fetal red blood cells. In pregnancy, this sensitization can be chemically prevented by administering anti-D immunoglobulin at specific intervals. But for chronically transfused patients, there is no drug that can stop the immune system from identifying and memorizing foreign minor antigens. Immune memory is permanent. Once these antibodies are formed in the body, precise genetic matching is the only clinical defense left. The danger compounds for highly alloimmunized patients who have been exposed to multiple mismatched units. They develop antibodies against several minor antigens simultaneously. To secure viable blood for these patients, laboratory technicians must physically cross-match samples, testing the patient's blood against individual donor units, tube by tube, to ensure that no hidden antigens trigger a reaction. Supplying a highly sensitized patient often requires hospitals to scour national registries, searching for rare donors whose specific ethnic backgrounds and genetic markers perfectly mirror the patient's own evolutionary history. Biological compatibility is written in the minor antigens, and evolutionary history the 8 major types ignore. Recruiting a diverse donor pool is a strict biological requirement. Treating blood as a simple 8-type system ignores the evolutionary history written on our cells, and it costs patients their lives.